Very good afternoon, students. So, in the last class, we focused regarding the significance of HMP pathway and its regulations, right? So, why the importance of NADPH? What is the importance of NADPH? That is why NADPH is required in order to maintain the erythrocyte membrane integrity and as well as to maintain the reduced form of the glutathione and as well as reactive oxygen species, right? So all those significance which we discussed. So now let us move on to the next pathway, that is glucuronic acid pathway, that is also called as uronic acid pathway. This pathway is an alternative oxidative pathway mainly for the glucose, sorry, and it is also known as glucuronic acid pathway because it is mainly concerned with the synthesis of glucuronic acids and as well as pentoses and even vitamin, that is ascorbic acid, that is vitamin C, can be produced or obtained from this pathway. So except in case of primates and guinea pigs, this pathway is going to, that is formation of this one intermediate is there in this 2 keto L gulonolactone formation. So that is not going to be formed in case of primates and guinea pigs. So this is also called as uronic acid pathway, right? So first of all, here the question which is come to our mind is, what is glucuronic acid? So this glucuronic acid is nothing but, which is obtained from the ancient Greek word, that is gluco means sweet, whereas uronic means urine, and it is a carboxylic acid, right? Glucuronic acid is a carboxylic acid. So its structure is very similar to that of the glucose molecule. However, this glucuronic acids, the sixth carbon atom of this glucuronic acid is mainly oxidized to a carboxylic acid. Sixth carbon atom of the glucuronic acid is getting carboxylated, or sorry, oxidized to form carboxylic acid. So its formula is going to be C6H12O7. Whereas the salts and the esters of this glucuronic acids are also known as glucuronates, that is going to be formed in this pathway, glucuronates. So the anion, that is anion of this glucuronate ion, should not be, where the glucuronic acid, you are not going to be getting confused with this gluconic acid. Just try to understand, glucuronic acid is totally different from gluconic acid. So the anion form of the glucuronates ion is nothing but that glucuronic acid, where it is going to be formed, and it should not be, you are not supposed to get confused with the gluconic acid. One more gluconic acid molecule is there. It is a linear carboxylic acid which is resulting from the oxidation of a different carbon of the glucose molecule. So this gluconic acid, which is a crystalline acid, and its formula is C6H12O7, mainly obtained from the oxidation of the glucose and used chiefly in cleaning the metals, as well as which is the source of its salts, from some of which are going to be used medicinally. Those salts are can also be used as a source from this in medical, for the medical purpose. So what is this glucuronic acid pathway? Mainly concentrated on which kind of reactions in this pathway? So let us see. Mainly the conversion of this UDP glucuronates to glucuronates, usually proceeding in this pathway by the way of formation of glucuronates 1 phosphate, which is a site for the short term regulation of this vitamin C molecule synthesis, mainly done by metapyrapone and other xenobiotic compounds, that is foreign substances or the foreign molecules, which is going to be isolated in the from the rat liver cells, that is hepatocytes. So the formation of this glucuronate molecule from the UDP glucuronate in case of the liver which is going to be extracted, and it is mainly enriched with ATP magnesium ion, but it is not going to be affected, it is not going to affect the formation of glucuronate 1-phosphate molecule. Understanding? So that is glucuronate 1-phosphate molecule is formed from the UDP glucuronate, or the conversion of glucuronate 1-phosphate to glucuronate. This and other particular information you can observe here. This is not an intermediate of this glucuronate formation from the UDP glucuronate, which is mainly suggested by this particular pathway. You can understand that the reaction is mainly catalyzed by the enzyme UDP glucuronidase enzyme. So this UDP glucuronidase enzyme is mainly present in the microsomal fractions, where its activity can also be stimulated by UDP N-acetyl glucosamine, which is mainly concerned with the particular stimulation. It is mainly known to stimulate UDP glucuronyl transferases, mainly by enhancing the transport of this UDP glucuronate molecule across the endoplasmic reticular membrane. Understanding? So this activity can be stimulated by UDP N-acetyl glucosamine, and it is going to stimulate this enzyme UDP glucuronyl transferase, mainly by enhancing the transport of this UDP glucuronate molecule across the endoplasmic reticular membrane. So this UDP glucuronidase and the UDP glucuronyl transferases, which is going to be similar, and it is very sensitive to the various detergents, mainly stimulated at lower concentrations and as well as generally inhibited at higher concentrations also. It is going to stimulate only at the lower concentration, and it can inhibit at the higher concentration level. The substrates for this glucuronidation reaction, which is mainly inhibited by UDP glucuronidase activity, which suggests that particularly primarily the suggestion was different. Now through this inhibition and as well as stimulation, it is suggesting that the later is contributed mainly by the UDP glucuronyl transferases. The inhibitors of this enzyme is beta glucuronidase and esterases, which is not going to affect the formation of this glucuronate, which is going to involve the formation of involvement, which is showing the involvement of this glucuronidation deglucuronidation reaction in this cycle. So this is all about this particular, what is this, glucuronic acid pathway. So the sensitivity of this UDP glucuronidase to metyrpone and as well as stimulatory xenobiotics, which was lost in the washed microsomes, which I explained you in the last slide. Even in the presence of this ATP magnesium, it could be restored by adding a heated liver high speed supernatant or even the coenzyme A molecule. So by this experiment, they have concluded that this glucuronate formation in the liver or the hepatocytes or the liver cells, which is mainly catalyzed by the UDP glucuronidase enzyme, and it is closely related to the UDP glucuronyl transferases. So this metyrapone and other xenobiotics, which is going to stimulate UDP glucuronidase enzyme by antagonizing the reaction, inhibition, which is mainly exerted indirectly by the combination of this ATP magnesium and as well as coenzyme A, both. So now let us see what is this formation and importance of this UDP glucuronate. So this glucose 6-phosphate is the molecule which is acting as a precursor in the formation of this ascorbic acid. So we can see here how this glucose 6-phosphate is undergoing or metabolizing in the liver cells. It is undergoing the reaction, liver microsomal fraction. So the glucose 6-phosphate is first getting converted to glucose 1-phosphate by the enzyme phosphoglucomutase, and here you are going to see the synthesis of the UDP glucose by the enzyme UDP glucose phosphorylase. This UDP glucose phosphorylase is also involved in the phosphorylation reaction, where UTP is added, inorganic pyrophosphate is going to be removed. And the next enzyme is UDP glucose dehydrogenase. It shows the dehydrogenase activity, where you can observe here oxidation reaction is going to happen that is UDP glucose to UDP glucuronate formation. And next will be the synthesis of ascorbic acid in case of some animals you can observe, not in case of guinea pigs and as well as primates. So this L-gluconate, which is going to be formed from UDP glucuronate, which is the precursor mainly for the synthesis of ascorbic acid. So in case of many animals you can observe this formation of ascorbic acid. So the enzyme L-gulonolactone oxidase, which is involved in the conversion of gluconate to ascorbic acid. So this L-gulonolactone oxidase is absent in case of man and other primates and the guinea pigs. So this vitamin C, which can be supplemented in the diet for these animals, where the absence of this enzyme, which can be observed in case of man, primates and guinea pigs, but directly we cannot synthesize this vitamin C molecule or the ascorbic acid. So it has to be supplemented through the diet in case of these animals. So how this oxidation of the L-gluconate molecule is going to happen, we will see. So first of all, this L-gluconate is going to be oxidized to 3-keto-gluconate and decarboxylated further to a pentose molecule, that is L-xylulose. Then this xylulose formation, 5-carbon molecule, is getting converted to D-xylulose. L form is getting converted to D-xylulose via xylitol by a reduction reaction, which is catalyzed by the what is that? Reductant molecule, NADPH molecule. So this is an NADPH dependent reaction which is followed by further oxidation, that is mainly involving the cofactor or coenzyme NAD plus dependent. So oxidation is going to happen, NAD plus dependent reaction, and reduction NADPH. Both is going to happen here in this reaction when the conversion of xylulose, L-xylulose to D-xylulose, which is happening via xylitol formation, right? So this formed D-xylulose, which is going to getting phosphorylated further, and it is going to enter into the that is exose monophosphate pathway or pentose phosphate pathway for the further metabolism reaction to happen. That's what we are observing in case of HMP, that is xylulose, formation of xylulose. So it can further involve in the other transketolase and transaldolase and transketolase reaction, what we have seen in the last classes, right? So further it is going to be metabolized. So you can see very clearly in this particular reactions, first structure, that is glucose 6-phosphate, is getting undergoing mutase reaction to form glucose 1-phosphate molecule. So you can see at the first carbon atom phosphate is getting attached, that is phosphorylation has happened, and then this glucose 1-phosphate, the shifting of the phosphate group from the 6th carbon atom to first, it has happened here. In the next second enzymatic reaction, the enzyme is a UDP glucose pyrophosphorylase. It is involved in the pyrophosphorylation reaction. That means UTP molecule is going to be incorporated, that is uridine triphosphate. That uridine triphosphate is getting incorporated to this glucose 1-phosphate to form inorganic pyrophosphate is going to be removed. So it forms uridine diphosphate glucose. So uridine diphosphate glucose is going to be formed. Then this uridine diphosphate glucose further undergoing dehydrogenation reaction. So as I said you earlier, so it is NAD plus dependent reaction here. So you can see here this NAD plus is acting as a cofactor to carry out this dehydrogenation reaction from uridine diphosphate glucose to formation of uridine glucuronate, sorry. So this uridine diphosphate glucuronate, which is going to be involved in the synthesis of glucuronides as well as protoglycans. Further it is undergoing hydrolysis reaction to form L-gluconate molecule. So then this D-gluconate molecule, then this further D-gluconate is getting undergoing dehydrogenation reaction, one more reaction, that is a reduction reaction, as I told you. So as well as it is undergoing dehydrogenation first to form this NADP plus is going to act as a cofactor. It is a reversible reaction. So you can make out in this glucuronic acid pathway, first reaction is reversible, second reaction is irreversible, third reaction is again reversible. So next reaction you can make out in this again a reversible reaction. So D-glucuronate to L-gluconate is going to be formed. So this reaction involves NADPH, which is going to act as a cofactor Further, it is involved in the removal of water molecule, that is dehydration reaction is happening, dehydratase enzyme. So then this resulting in the formation of L-gulonolactone. So then this L-gulonolactone is further undergoing oxidation reaction. So this particular reaction involves reaction can be observed in many animals except in humans, primates and guinea pigs, because of the lack of this particular enzyme here, L-gulonolactonase. So then this resulting in the formation of 2-keto L-gulonolactone. So this 2-keto L-gulonolactone is further getting converted to ascorbic acid. So it is involved in the synthesis of ascorbic acid further. So where in case of other animal, man, and in case of primates, guinea pigs, this should be supplied through the diet. This ascorbic acid cannot be synthesized, right? So here, from here also, D-glucuronate to L-glucuronate can be observed, and formation of xylulose, and through the formation of xylitol, L-xylulose to D-xylulose, then it can also be involved in the synthesis of this ascorbic acid. So next, coming for the topic, importance of this glucuronic acid. So what is the importance mainly of this glucuronic acid in order to why is it so, why this pathway should happen, what is the importance? So it provides mainly UDP glucuronic acid in the active form of the glucuronic acid. That is, UDP glucuronic acid is going to be obtained in the active form of the glucuronic acid, mainly conjugation of the bilirubin, and mainly by the conjugation of steroids and conjugation of the various drugs, which will make this glucuronic acid as a more water soluble, and it makes very easy to excrete out from the body. And it is also involved in the synthesis of glycosamine glycons. As we have seen, it is also involved in synthesis of glycosamine and glycons, glucuronides, as well as your protoglycans, all these things which we can see. So that's why this glucuronic acid plays a very important role in the synthesis of these molecules, which can be conjugated with these molecules and even involved in the synthesis of other molecules. So what is the effect of this drug on this uronic acid pathway or the glucuronic acid pathway? So the administration of this drug, especially barbitol as well as chlorobutanol, so these two are the drugs which are going to increase the uronic acid pathway in order to achieve more synthesis of this glucuronate formation, mainly from the glucose molecule. So it is going to act as a inducer to increase the uronic acid molecule, formation of glucuronic acid, the uronic acid, which will help you in the synthesis of glucuronate molecule from the glucose molecule. So other certain drugs like aminopyrin as well as antipyrin, which is also found in order to enhance the synthesis of these ascorbic acid in case of the rats. So coming for this pentosuria, how this pentosuria is going to happen? So it is a rare genetic disorder which is mainly occurring due to the deficiency of the enzyme NADP plus dependent enzyme, that is xylitol dehydrogenase. So this xylitol dehydrogenase enzyme is deficient, then it is going to result in the formation of this pentosuria, because because of this deficiency xylitol dehydrogenase enzyme, it will not be involved in the conversion of L-xylulose to xylitol. So L-xylulose cannot be converted to xylitol. So this is via xylitol you can also see the formation of D-xylulose. So L-xylulose cannot be converted to xylitol molecule. So the affected individual who is not going to excrete any large amounts of this xylulose in the urine, and it gives a very positive Benedict's test. So from this Benedict test we can confirm that the patient is suffering from this pentosuria because of this enzyme deficient, that is xylitol dehydrogenase. It cannot excrete this xylulose molecule in the urine. So particularly, so the xylulose cannot be further converted. So you can see the affected individual excretes the large amounts of this xylulose only, no xylitol formation in the urine. We can see only xylulose. So this can be achieved, or this can be tested mainly by doing this Benedict's test. So what is this essential pentosuria particular symptom. It is a asymptomatic in nature. It is going to where you can come across this barbiturates and aminopyrine, which is going to induce this uronic acid pathway, and it will also increase the excretion of L-xylulose. So during this condition, in pentosuria condition, the patient with this pentosuria, you can give this barbiturates and aminopyrine, which will induce this particular pathway in order to increase the excretion of more amount of sorry, this barbiturates and aminopyrine is going to act as a inducer molecule. So this thereby it is going to increase this pathway and increase the excretion of more amount of L-xylulose in the urine, in this case of the patient who is affected with the xylitol dehydrogenase enzyme activity. Is it clear? Okay. In the next class, we will move on to the next pathway, that is Entner-Dyatlov pathway, Entner-Dyatlov pathway, that is ED pathway, and other reactions with respect to this ED pathway. Thank you.